Hey there. Let's talk briefly about cell division and DNA replication. And we're going to introduce this topic by thinking about human development starting from a single cell. Let's think about you and how you came to be here as an adult human being. You began when two cells fused together to make a single cell. These are human sperm cells shown under a microscope. Each one was produced by a type of cell division called meiosis, in which half the genome is inherited by the daughter cells. So most human cells have 46 chromosomes, but these cells have 23, half of that number. The human egg was also produced by meiosis and also has 23 chromosomes. Here's a sperm cell, which most likely had 23 chromosomes from the biological father, and it's fertilizing this egg cell, which also most likely had 23 chromosomes from the biological mother. The cell that results from the union of egg and sperm is called a zygote, and the zygote has 46 chromosomes if it's a human zygote. So this zygote, the cell, has 6 billion base pairs and somewhere around 20,000 protein-coding genes, give or take, distributed across these 46 chromosomes. But humans aren't just a single cell, of course. Adult humans probably have somewhere around 37 trillion cells. That's the best estimate anyway at the time this video is made. It could change. Okay, so we went from one cell to 37 trillion cells. So that zygote had to divide into two cells, and then those two cells had to divide into four cells, and so on, until you reached your adult size. Now we've learned that cells have a genome that's made up of DNA. And before the zygote was able to divide, it had to copy this genome and divide the DNA evenly between the two daughter cells. Then each of the cells had to copy their genome and divide the DNA evenly, and so forth. So now here you are with about 37 trillion cells, and just about every one of these cells has a copy of that whole genome. Now, by the way, growth and development aren't the only reasons that cells divide. They might also divide for tissue regeneration or wound repair, and cells also divide for the purpose of reproduction. Some organisms reproduce asexually. They simply copy their genome, then divide into two identical cells, such as these E. coli cells. Other organisms like humans reproduce sexually, and we produce the egg cells and sperm cells to do that, using that special type of cell division we mentioned called meiosis. But no matter what the reason is, the essential thing is that a cell needs to do this before dividing. It needs to copy or replicate its DNA so that each daughter cell has the entire genome, which is necessary to maintain life. We can't just give the daughter cells half of the DNA and then have them give their daughter cells half of their DNA and so on, because, well, we'd run out of DNA pretty quickly. Not only that, we'd be missing a lot of really key genes in some of the daughter cells, and the daughter cells would not be able to make all of the proteins that they would need in order to metabolize. So the cell will copy its DNA first to then give one copy to each of the two daughter cells. DNA replication takes place in prokaryotes and eukaryotes, and some of the details of how that happens are different between those two types of organisms. But what is the same is that enzymes called DNA polymerases catalyze the synthesis of new DNA molecules, and they do so according to the base pair rules that we've already learned. There are a bunch of different DNA polymerases, but we'll just keep this simple because they share the function of making new DNA strands, using old ones as a template. DNA polymerase adds new nucleotides to the 3' end of a newly forming strand. It takes a nucleotide triphosphate, or a nucleotide with 3 phosphate groups, in other words. In this case, it's a nucleotide with thymine, and it joins it to the 3' carbon of the nucleotide that is already there. In this case, it's a cytosine nucleotide. If DNA polymerase tries to add the incorrect nucleotide, it usually won't work because the hydrogen bonding isn't correct. But occasionally the wrong nucleotide is added. Many DNA polymerase enzymes, including the main DNA polymerases that elongate new DNA strands in human cells, can correct mistakes by excising the incorrect nucleotide and then adding the correct one. We call this proofreading. Let's use the base pairing rules to predict the complementary strand of DNA. Here's a sequence of DNA. This is part of a gene. Let's read it from 3' to 5', TTC, TGA, AG, AC. Now let's predict the sequence of the other strand. First of all, let's determine polarity. Remember that the second strand is antiparallel, so it's going to run 5' to 3'. Now we're just going to use the base pairing rules. DNA polymerase will add an A to match with T, then another A, then G because C pairs with G, then A, C, T, T, C, T, G. Now we just do that for the whole genome, and then we have all of the DNA that we need to divide. Now, I've made that look simple, and it's not. There are many enzymes involved, and the process of DNA replication is super complicated and fascinating. But for now, we can move forward knowing that cells must replicate their genomes before they're able to divide, and that DNA polymerase adds new bases to make new strands of DNA using the complementary base pair rules. Adenine and thymine pair together, and cytosine and guanine pair together. All right, see you next time.